presentations end. Thank you so much. So we'll stop the recording after the presentations have finished. I want to welcome you all and thank you all for participating in the Synthetic Drug Subworking Group Coalition meetings. I wanted to share that this is the first hour of 4 hours. You are welcome to stay for as many of the sessions as you would like. We are starting off with the 2 sections on manufacturing and trafficking over the next 2 hours, followed by the 2 sub-working groups on detecting threats and emerging patterns. And then tomorrow we will have 3 sessions on public health at the same time. At the bottom right hand, you see Synthetics Coalition at state.gov. The State Department is assisting with secretarial duties, so feel free to reach out to us with any questions, concerns, comments, and so forth. I want to also share that registration for the working groups and sub-working groups will remain open for the length of the coalition till September of next year when we end. So feel free to share these links with other colleagues to register for the sub-working groups of the coalition. Again, if you need the links, we can send them to you digitally if you email us at the address below. A new coalition website is also under development, and the presentations from today will be recorded and posted on the website. But as I mentioned earlier, not the commentary, not the comments period that will come later on today. International organization coordinators that are volunteering in supporting us will also prepare a summary of the meeting, and so we will share that with all of you as well. Now, in the next few weeks, the Secretariat will be sending the meeting schedule and calendar invites to all participants for the next few months through February of next year. So you will have all of the full schedule for the year, the dates of the meetings, and then you'll also have the calendar invites for the next few months. And finally, I wanted to highlight that for this sub-working group 1.1, manufacturing, we currently do not have a sub-working group co-chairs that have been identified yet. So we are reaching out to all of you and inviting you, if you would like to co-chair this sub-working group 1.1. Again, please email us at syntheticscoalition@state.gov. We have the opportunity for both 1.1 as well as 2.1 on data collection, analysis, and trends to recruit co-chairs. So today we'll have provisional co-chairs for those sub-working groups that I will discuss in a moment. I want to end by sharing the meeting schedule for the next sessions. So this is the October session, which is Framing the Issue, where we talk about the challenges and the issues of the day related to this topic. Then between November and February, we'll be looking at sharing best practices and models and finding intervention points for activity initiatives, as well as making deliverables and commitments as a result of these meetings. Then in March, we will have a midterm check-in with the co-chairs of the working groups to take stock of the progress that we've made. And that will be followed by action planning and implementation of the sub-working groups between April and July, followed by evaluation and transition, looking at the sustainability of our work for years to come. However, the coalition's work will end with a final working group meeting in September. So we'll be spinning off initiatives, projects into other organizations and other initiatives that are already under existence or that we will initiate through the coalition's work. So with that, I'm going to stop sharing my screen, and I am going to introduce first the international organization coordinator that's assisting us with all of the support related to this sub-working group. His name is Rainer Punks. He's the program manager for drugs and precursor chemicals at the UNODC Regional Office for Southeast Asia and the Pacific in Bangkok. And I'll just pause a moment so that he can introduce himself and say hello. I'm not sure if Ryan.
Good morning.
Thank you, Ryan.
Yeah, good morning, everyone. Good to see you, and that I got the right link now. And yeah, I'm still reaching out to the co-chairs that have been invited, but I don't know, some are on leave, others need their, as we mentioned in the first meeting, they need their approval from the higher ups, and that takes time. So as soon as I get any reply, we'll let you know. And we'll be taking notes here and see how far we can go. Thank you.
Thank you. Thank you so much, Rainer. Very much appreciate your efforts. Now I want to introduce our 2 provisional co-chairs for today, who are familiar faces if you attended the working group meetings in September. We have Greg Williams, the Detective Superintendent from the National Organized Crime Group from New Zealand Police, and Bill Solly, the INL Director from U.S. Embassy Vientiane in Laos. So I'll turn it over to them for their welcome as well, and then they will proceed with the meeting and introduce the speaker for today. Thank you.
Shall I go first? Kia ora team, Greg Williams here. Pleasure to sort of step into the chair in the prelim for this very important working group. When I was up at the IDEC meeting in Jamaica talking to Bill Kimball, who's sitting in the chair, co-chair on the other side of the world, I guess we both agreed that this particular working group sitting around precursors and the manufacturing of meth is probably one of the most critical in this coalition, along with the focus on victims. So very happy to be as part of this.
Everybody, my name's William or Bill Solly. I'm the Director of International Narcotics and Law Enforcement Affairs in U.S. Embassy Vientiane in Laos. It's great to be here, even if I'm just filling in. It's nice, Greg, nice to see you again. And I think without further ado, we should go ahead and turn it over to our presenters today. I believe we're starting out with Justice Teddy, the Chief of the Drugs Laboratory and Scientific Services Branch, Division for Policy Analysis and Public Affairs at UNODC.
Synthetic drugs present a unique challenge to international drug control, which seeks to preserve the health of a global community. Unlike the production of plant-derived drugs, such as cocaine and heroin, which are geographically constrained, synthetic drug manufacture can occur anywhere, depending on human creativity and a few key chemicals. This fluidity makes production extremely mobile, and any country a potential source. Yet, this is not necessarily a totally new phenomenon. For over a century, international drug control has had to contend with waves of emergence of synthetic drugs. Derivatives of morphine in the 1920s, fentanyl analogs in the 1980s, and amphetamine-type stimulants in the 1990s. The 2010s marked a defining period for synthetic drugs, with the unprecedented emergence of hundreds of substances which mimic the effects of drugs harmful to human health and therefore under international control. Substances commonly referred to as new psychoactive substances have been reported in 141 countries and territories worldwide. And as of today, there are over 1,230 substances reported. For unprepared societies, the effects of these cyclical surges of synthetic drugs had severe consequences. But none of these events has been as distressing as the ongoing synthetic opioids crisis affecting mainly North America with fentanyl and its analogs, and parts of Africa, Asia, and the Middle East with tramadol. More specifically, in 2023, the World Drug Report identified that methamphetamine has emerged as a prevalent synthetic drug, with manufacture and use expanding in Southeast Asia, North America, Southwest Asia, Africa, and Europe, with seizures tripling in just 6 years. A growing number of laboratories have been detected in Central Asia, Southeast Asia, the Near and Middle East, Africa, Europe, and North America, producing a range of stimulants, depressants, and other novel dissociatives. Synthetic opioids have claimed tens of thousands of lives in North America through overdoses, the majority associated with fentanyl. Synthetic drugs have also emerged as a threat in fragile and conflict-affected areas, from Afghanistan to Lebanon, to Myanmar, to Syria, to Ukraine. In Latin America, the markets for MDMA and ketamine are growing. Africa continues to struggle with the non-medical use of tramadol. The Middle East is flooded with captagon pills, and ketamine is becoming a mainstream drug in some population groups, and seizures are hitting record levels in Southeast Asia. What makes synthetic drug manufacture so attractive? The ability to chemically synthesize immensely potent drugs at a low cost has opened doors to huge profits for organized criminal groups. Clandestine manufacturing laboratories can be set up close to markets, thus shortening the supply chains. Synthetic drugs are cheap and fast to produce. Manufacture can be more discreet and easier for traffickers to conceal. To effectively respond to this global threat, it is important to first understand the multiple phases involved in synthetic drug manufacture, recognize the challenges facing traditional supply reduction efforts to effectively detect and interrupt such manufacture. Now let's have a closer look at what qualifies as a synthetic drug. How are they manufactured? And where are the opportunities to disrupt such manufacture? It is hard to take action or talk about effective responses if we do not understand the chemistry behind synthetic drug manufacture. The capacity of forensic drug analysis laboratories to provide verified information on chemicals found within synthetic drug samples is essential. The rapid evolution of synthetic drug markets has marked a turning point for drug and precursor identification. Analyzing and identifying a large number of unknown and sometimes structurally related substances without ambiguity has become much more complex. High-potency synthetic drugs also pose a severe safety risk for anyone interacting with such drugs. We need to look at how we can strengthen the capacity of frontline officers and forensic personnel to safely handle and minimize the risk of exposure to these highly toxic substances, some of which are unknown till they are identified. Let us not forget that precursor chemicals are essential to the manufacture of synthetic drugs. If there are no precursor chemicals, there can be no synthetic drugs produced. Illicit manufacturers are circumventing the use of internationally controlled substances through minor modifications to synthesis pathways and the use of non-controlled designer precursors to make these substances. Online discussion forums, through both the clear and dark net, present hidden platforms for illicit manufacturers to exchange information and know-how on designer precursors, pre-precursors, and new synthesis pathways for the production of novel synthetic drugs. In order to effectively and safely respond to these complex challenges, it is crucial that national, regional, and international drug control efforts and policy decisions are guided by science. Secondly, international trade in drug manufacturing equipment and materials are regulated. Thirdly, collaborations with private sector and industry are strengthened, and the capacity of countries to implement international drug control scheduling decisions is enhanced. The manufacture of synthetic drugs does not require control over large agricultural areas and the local population to grow drug-related crops. Clandestine laboratories used to manufacture synthetic drugs are comparatively small in size, and the chemicals and equipment needed can be bought under the guise of licit business. This makes synthetic drugs a particularly attractive source of income for insurgents and non-state armed groups. Clandestine laboratories are discreet, often integrated in urban settings, and can be set up easily, disbanded, and relocated. This renders clandestine laboratories harder to identify, trace, and track. Their presence in urban settings makes it all the more difficult and dangerous to safely dismantle if not guided by science. How can clandestine laboratories be identified and safely dismantled? In order to identify clandestine laboratories, it is crucial to first recognize common signs of illegal activity associated with clandestine laboratories. We should be able to pinpoint typical locations for clandestine laboratories in both urban and remote settings. We should be able to understand key components of what formulates a clandestine laboratory from the chemicals and equipment needed. Another important aspect of effectively addressing synthetic drug manufacture is the disposal of chemicals used in their illicit manufacture. Seized chemicals, when not correctly identified and managed properly, can cause serious and immediate risk to human health, communities, the environment, and critical resources such as groundwater, drinking water, and agricultural land. It is therefore important that chemical disposal operations are conducted in a manner that seeks to reduce or eliminate this risk. A significant risk to drug control efforts is the potential for reintroduction or reintegration of these seized materials into the illicit drug market, often due to the lack of well-defined in-country measures for secure storage, as well as timely and efficient disposal. This is often the result of the absence of legislative provisions for pre-trial disposal of seized materials, the lack of suitably trained personnel for safely handling, transporting, and disposing of chemicals, the lack of infrastructure for safe housing of chemicals prior to disposal. It could also be due to low national forensic capacity for unambiguous identification of the chemicals, which is a prerequisite to the selection of safe disposal methods. It could also be due to the absence of in-country operational strategies and/or lack of interagency coordination in conducting these technical and often delicate operations. Quite often, ill-informed acquisition of very expensive incinerators as a quick fix solution to the disposal of seized chemicals have turned out to be unsustainable and costly. With synthetic drug manufacture, we are facing multiple challenges which require international cooperation to bridge information, capacity, and resource gaps in order to be able to address the synthetic drug problem. We need to target the supply chain as a connected whole, to improve monitoring and control measures, to be more prudent in regulating access to chemicals, and to collect data on a global scale to better understand the flows and the actors involved. Swift exchange of accurate scientific information across borders is also essential, as well as improving global capacities to keep up with developments in illicit manufacture. Ladies and gentlemen, the challenges ahead of us are immense and require international cooperation and coordination to step up global action on the challenges of synthetic drugs. This is the premise behind the global coalition. I am confident that discussions from this sub-working group over the coming 4 months will provide important insights and highlight solutions from around the world that can enhance responses to manufacturing of synthetic drugs and their precursors, a key component of a comprehensive response. I thank you.
Okay, well, I thought that was a really excellent presentation framing the challenges as well as the trends affecting this issue. I think before we move into the discussion period, we will hear from Emily Lockhart, the Senior Forensic Chemist at the Drug Enforcement Administration Special Testing Laboratory.
Hello, everyone. Thank you for having me today. I do have some slides I'm going to share, so hopefully I'll get those up smoothly and easily.
And we're confirming that we can see your slides. Thank you.
Wonderful, wonderful. All right, so thank you for the opportunity to be here with you guys today. The variety of synthetic drugs on the illicit drug market and the plethora of chemicals used to make those drugs truly does face law enforcement and forensic laboratories with an immense challenge these days. So I'm really excited to be here to talk about it and hopefully come up with some ideas of what we can do to combat this a little bit. So I am going to be presenting from more of a US perspective. So here in the US, we're primarily focused on synthetic opioids. namely fentanyl, but we do have some other ones that I'll talk about a little bit throughout the presentation. Novel benzodiazepines and xylazine are really the other big synthetic drugs that we're looking at right now. And towards the end, as I am a forensic chemist, I'm going to talk a little bit about some of the challenges the forensic laboratory faces. So this graph here shows you the evidence submitted to the DEA laboratory system by suspected drugs. So this is what the law enforcement officer has listed the substance to be. And you can see that about half of the evidence that we receive in the DEA laboratories is either suspected methamphetamine or suspected fentanyl or fentanyl-related compounds. So primarily, half of what DEA is looking at are these two main classes of drugs. Here in the US, most of the methamphetamine is coming from Mexico. They're producing really high quality D-methamphetamine hydrochloride, typically in crystalline form. So there's not much need for a lot of domestic synthesis of methamphetamine here in the United States. Generally, if we do see methamphetamine clan labs, it's on a much smaller kind of user-based scale. So here's a look at the new synthetic opioids that we have detected in the DEA laboratory system for the first half of 2023. So you'll see that by far fentanyl is really the main substance that we're concerned about in this category, as it represents 81% of the opioid identifications. Now this group doesn't include other synthetic opioids like hydrocodone or oxycodone, things like that. This is looking more at this kind of new category of substances. Parafluorofentanyl is another fentanyl-related substance that we're really trying to keep an eye on right now. Most commonly, we see fentanyl and fluorofentanyl together in the same mixture. So that's really interesting as well. Let's see, parafluorovalerofentanyl and really long chemical name here, T-butyl 4,2-methylphenyl aminopyridine 1 carboxylate, which is that very top one on the graph there, were both identified for the first time in 2023. So those are some new substances. We believe that long chemical name is a byproduct of the synthesis of fentanyl that was detected in the final product. But one of the things we're really monitoring closely right now is the emergence of the nitazines. So you'll see in my list here, we do have several of them that we've detected in the laboratory. And I have another slide in my slide deck to kind of go more in detail about what we're looking at with those. You'll also see that in this list, there's a number of fentanyl precursor chemicals. So all these Bach compounds, the Bach 4-piperidone, Bach 4-AP, and the fluorinated version, as well as things like aniline, 2-phenethylbromide are also monitored in this list. So we're starting to keep track of more precursor chemicals as well in the laboratory. So we don't have a lot of domestic synthesis of fentanyl in the United States. We're very much more of a user country as opposed to a source country for most illicit drugs. But while we don't have a lot of actual synthesis of fentanyl here in the United States, we do have a lot of milling operations or adulteration facilities where they're cutting the fentanyl with other substances, whether it's other controlled substances or diluents to make the product bulkier. We also have a lot of tableting operations here in the United States where they get the fentanyl in powder form and then convert it into tablets. So here we're going to look a little more specifically at the nitazines. Nitazines were initially developed as part of pharmaceutical research by a Swiss chemical company. And much like other NPS, there's a core molecule that can be changed in certain positions to make a whole series of different compounds. So you can see there in the chart, in the table, the list of nitazines that we've identified in the DEA laboratory system. And this is an ever-growing list. We have a few other ones that we're monitoring that haven't actually been seen in samples yet. And most commonly, these are seen in powder form, and often we'll see them mixed with other nitazines, so multiple nitazines in the same sample. And the bar chart there shows you some of the other substances that are co-identified with these nitazines. So seeing combinations of fentanyl and other opioids, or even the benzodiazepines with the nitazines is more and more common. The second class after opioids that we're looking at are the novel benzodiazepines. Historically, so going back maybe 5 years or so, atizolam was really the main new benzo that we've been monitoring. It's kind of the original one we saw in the illicit drug market. After that, we started seeing more clonazepam and flualprazolam, but in 2023, bromazolam has really emerged to be the front runner, the most popular benzodiazepine we're seeing in the laboratory system right now. The last substance I'm going to talk to you about is xylazine, which has become more prevalent here in the United States. It is a non-controlled opioid analgesic, and in the United States, it's only approved for veterinary use. So they use it during animal surgeries. We believe that we see fentanyl adulterated with xylazine to help exaggerate the analgesic and euphoric effects of the fentanyl, as well as lengthening the duration of the high, as xylazine is longer acting than fentanyl is. Xylazine was first seen in heroin samples by the Heroin Signature Program at my laboratory back in 2000. So it's certainly not something that is new, but it has only recently emerged in fentanyl samples. And the maps I have here show the spread of xylazine from the Northeast part of the United States in 2019 through present day. So far, there are only 2 states that have not seen xylazine in fentanyl samples. So it is pretty widespread here in the United States. All right, so moving to forensic challenges. The assortment of synthetic drugs that are on the market pose a significant challenge to forensic laboratories. Often these substances are first encountered by customs officers or officers on the front line that are using field testing or presumptive testing to initially make some sort of identification or decision about whether to seize the material. The complexity of opioid mixtures and the difficulty in maintaining up-to-date libraries on portable instrumentation can really be a challenge. It does require a lot of coordination between the frontline officers and the laboratory to ensure that the officers have what they need to make informed decisions. Reference materials are the key to the identification of any controlled substance. And a lot of times reference materials for new materials can be either unavailable or prohibitively expensive. And that poses a challenge, not just for laboratories in developing countries, but a lot of times it's really expensive for even, the biggest laboratories to afford some of these materials. When a new substance is detected and reference materials not available, the structural elucidation process is needed to fully characterize what that substance is. And that can be a really time-consuming process. A lot of laboratories don't have the necessary equipment to do that or the necessary expertise, as that's not something that typically forensic chemists are trained to do. After the material is characterized and the reference material is either synthesized or purchased, then it needs to be added to the laboratory's validated methodology to show that the techniques that are available in the laboratory can sufficiently identify that substance. Many substances in both new and traditional drugs are unstable, meaning that they degrade under certain conditions, whether that's heat or an acidic environment or a basic environment. In these instances, specialized sample preparation or alternative instrumentation may be needed for analysis, and that's just another cost that the laboratory faces, just so that they can successfully identify some of these compounds. So while new synthetic opioids like nitazines are unlikely to replace fentanyl in the United States, they will likely continue to be encountered in seized drug exhibits. Justice alluded earlier in his presentation about the number of overdoses here in the United States. Some of these nitazines are even more potent than fentanyl. So they do require numerous doses of naloxone to successfully revive, somebody experiencing an adverse health event. And substances like xylazine don't even interact with the opioid receptor. So naloxone is not effective at all against a xylazine overdose. The variety of novel benzodiazepines will continue to be detected in seized drug exhibits, and new ones will continue to emerge as older ones are regulated. Xylazine's presence, at least here in the United States, will continue as long as it remains unregulated and is readily available. We are looking at potential alternatives that may be selected should the supply be reduced or xylazine be regulated. And forensic laboratories must provide comprehensive testing to detect the emergence of new substances in a timely manner. And ultimately, the collaboration and communication between forensic laboratories is going to be key to make sure that new substances are detected in a timely manner and the data is shared within the community. And that's all I have. So I'm happy to take any questions, and I'm looking forward to the discussion that's going to occur. Thank you.
Thanks very much, Emily, for your really fascinating presentation. Although both of these presentations, revealed the depth of the problem, I hope